Last time I see you today, and then tomorrow morning, I think at 10, I have to check. It's the last time I'll see you guys for a while, so feel free to contact me by any of those routes, and I'm more than willing to help everyone. All right, antibody. How many folks, just so I can get oriented here, how many folks have heard the word antibody and sort of have some idea what it might be? Kind of, a little bit. Everyone. Good. Good. So that means either the ones who are most interested showed up, and most knowledgeable showed up, or you have actually heard about antibody. So, in circulation, we have antibodies in circulation. We'll hear a great deal about them over the next couple minutes. Now the word different, I have to define the word different, but I'm going to ask you a question. Do we have 100 different antibodies in circulation? 1,000? 1,000,000? 1,000,000,000,000? That's the question I'm going to ask you guys, but I have to define the word different. Different means they're specific, what they recognize. I mean, everything in the immune system that I've been trying to teach you guys is that the immune system is about, it needs to recognize foreign, and it needs to recognize foreign and distinguish it from itself. We haven't gotten into autoimmunity yet, and I'm actually not teaching the autoimmunity class this year, so that's yet to come. But the immune system works by recognition. Okay, so different, when I say different antibodies, I'm talking about antibodies that recognize different things. And things being a nice, important scientific term. So, do we have 100, 1,000, 1,000,000, or 1,000,000,000,000 different antibodies in circulation right now? How many folks think we have 100? No. How many folks think we have 1,000? One person. How many folks think we have 1,000,000? About half. How many folks think we have 1,000,000,000,000? The other half. I forgot 1,000,000,000. Isn't a billion in there somewhere? Billions between 1,000,000 and 1,000,000,000. So it ends up we have 1,000,000,000,000. We had 10 to the 11th different antibodies in circulation right now, each of us. And those 10 to the 11th different antibodies can see 10 to the 11th different molecular signatures. We'll define the scientific term for molecular signature in a moment. But those antibodies, 10 to the 11th, can see different chemical structures. Okay. Now, let's go back a second and ask, does anyone know what the macromolecule that makes up an antibody is? What is the macromolecule for an antibody?
Is it B cell?
Excuse me?
B cell?
No, that's a cell. What's the macromolecule? Macromolecule means DNA, carbohydrate, lipid, protein. That's what I mean by macromolecule.
Protein.
Protein. So antibodies are proteins. Okay. So if I give you 10 different proteins from the human body, how many different genes are associated with those 10 different proteins? In general, there are always exceptions. There's no way about the exceptions. How many genes? So I hand you 10 different proteins, and I say, tell me a little bit about the genetics of these proteins. How many different genes are we talking about? I thought you said. Oh, you didn't say. I know she said, but I thought you said something. No. So you say 10.
Usually it's one gene, one protein component.
Usually one gene, one protein. Do you guys buy that? Usually one gene, one protein. How many folks don't buy it? I saw a couple of people shaking their heads. Okay, why do you not buy it?
Multiple genes could code for protein, the same protein.
Like, is there something you were thinking of in particular?
I was thinking of like how there's multiple codons for the same amino acid sequence.
Correct. Yeah, you also said 10 was the wrong number. What were you thinking? I'm not ignoring you. I'm trying to put together what you guys are thinking.
So like when we were in our program, there are repressors and like different types of regulators that are coded by different
Right, but those operons, operons are mostly bacterial, not human, but operons make repressors and inducers, but those repressor inducers are also proteins. So if there's a gene for a repressor, there's a gene for a repressor. If there's a gene for an inducer, there's a gene for an inducer. So even though the operon makes has a series of genes that lead to a metabolic process, the repressors and inducers associated with that are proteins too, and they have their own genes.
Would it have to do with,
Hold on 1 second. There was one, you said 10 was the wrong number also.
I was thinking specifically exon splicing, so one gene could make many different proteins due to how they're spliced
The
together.
splicing is a problem. We'll get to plus splicing in about an hour or so. Ignoring splicing. So, in general, when I said, I tried to make this somewhat generic by saying in general, one gene, one protein. You have a stretch of DNA, you have the triplets or the codons that you're talking about, and these codons will code for one amino acid, this codon codes for the next amino acid, next one, and that chunk of 100 amino acids, 1,000 amino acids, whatever, is going to be a protein, and that's one gene associated with it. Sure, we have alternate splicing, we have operons, we have isoforms of proteins. But in general, one gene, one protein. Okay, how many genes in the human body? How many genes make up us? Again, ignoring a little bit of splicing and some of the subtlety, there's a number that most people would throw out.
20,000?
Does anyone know? You may not know. I mean, this is not something you know. Actually, excuse me. Actually, people should have read this, like, the New York Times would think there'd be articles on it, as well as obviously the scientific book. Okay, so let's throw out some numbers.
He said it.
Like 20,000?
20,000, perfect. That is actually the number that I use. It's 20 to 24,000. So there are 20,000 genes in the human body, which means about 20,000 proteins, give or take. Even 40,000, a couple of alternate splices, a couple of isoforms, or whatever, maybe 30,000 proteins. 20,000 genes, 20,000, 30,000 proteins. Let's circle back. How many antibodies did I say you had in circulation? Trillions. Each antibody is a protein. So you need trillions of genes to code for antibodies. Each antibody has a different specificity, which means its amino acid sequence is going to have to be different. Otherwise, if the amino acid sequence of this antibody and this antibody are absolutely identical, they're going to bind to the same molecule. So the amino acid sequences here and the amino acid sequences here have to be a little bit different. So we need a gene to code for these amino acids. To make antibody 1, we need a gene to code for these amino acids. To make antibody 2, we need a third gene to code for antibody 3, up to a trillion. So, major problem. We do not have trillions of genes, yet we have trillions of proteins that are called antibodies that are all different. So the challenge for today is, how do we do it? How do you modify the genetics of the human host, or what molecular biology tricks are there out there to create trillions of different antibodies from a very small genome? So that's part of the challenge for today. But let me first introduce you to an antibody. Antibodies, excuse me, antibodies are also called immunoglobulins. You'll see IG, capital I, little g for immunoglobulin. It's another word. You may have heard, this is old science, but you may have heard gamma globulin. You may have been, when you were a kid, you might have heard that someone got a gamma globulin shot when they were traveling to a foreign country or something. So where does that word globulin come from, and gamma in particular? Ends up, if you take human serum, remember I said the antibodies are in circulation in blood. Take human serum and you run a column that separates by size. You're just separating proteins by size, and they got peaks like this. Small to larger. And albumin is a protein in the serum called albumin, which is the most predominant single protein in the body. And then there was an alpha peak, a beta peak, a gamma peak. The gamma peak ends up going to be antibodies in the end of the day, but not when they did this experiment. And they were globular-shaped proteins, because this column, this technique that doesn't matter what's going on here, this technique separates by their structure as well as their size. So these were globular-shaped proteins that came out in the gamma fraction. Hence they were initially called gamma globulins, and still we call them immunoglobulins. After a lot of work, we found out they ended up looking like this. So, an antibody, an immunoglobulin, is actually 4 chains. So we already have a problem, a factor of 2. With that 20,000, now it's 10,000, because there's got to be a gene for what's called the pale blue is the light chain. Well, there's a heavy chain, let me change. There's a heavy chain, it's bigger, and a light chain, it's smaller. So a heavy chain and a light chain. So we need a gene for the heavy chain, we need a gene for the light chain. So already now the trillion genes that we need 2 trillion genes. Because we need a heavy chain and a light chain gene. Okay. So, an antibody is made up of 2 heavy chains, here and here, 2 light chains here and here. The amino acid sequence for the light chains are identical to each other. The amino acid sequences in a given antibody. The amino acid sequences for the heavy chain, for the 2 heavy chains in a single antibody, are identical. And as you can tell, it's Y-shaped, and I'm going to be standing like this a lot in class today, so you can get used to it now. So, an antibody has a trunk and 2 arms that are identical. The trunk is identical, the amino acid sequence here is identical to the amino acid sequence here, and the two polypeptide chains that make up the 2 arms of the antibody are identical, light to light, heavy to heavy. Okay. The antibody has 2 functions. One function is it must recognize the foreign agent. That's what antibodies do, they recognize things. And then they must do something with that foreign agent in order to protect us. So, the foreign agent binding part of the antibody is called the antigen binding. I haven't defined antigen yet, but I will in a minute. So you'll notice the arms are the antigen binding component of this antibody. And the trunk, this part, is giving effector activity. What those translate to mean is, here's the antibody. The antibody grabs something foreign using its 2 arms. This part of the antibody communicates with the rest of the immune system to do something once the antibody has grabbed it. You might remember complement from a couple of days ago. What's today, Monday? From last Wednesday. Complement, the classic pathway of complement, has antibody bound to the bacteria. Oh, I meant to look, I promised I would look this up, and maybe this will be an exam question since only you guys will get it right. I talked about what does complement kill. Complement can kill bacteria, and complement can kill fungi. So I'll put that on the exam, and then only 20 of you will get it right. Thank you for coming. So remember, the classic pathway, antibody binds the bacteria, or the pathogen now, I have to say pathogen because it's fungi as well. And it grabs the pathogen with its 2 arms. So looking at me as an antibody again, you can grab a bacteria here and here. And then this part of the antibody is sticking out in solution. The next thing that happens in the classic pathway is C1q binds to the antibody. It ends up, C1q is recognizing the trunk, this portion of the antibody, the effector region. So you can fix complement with an antibody because the trunk of the antibody is the effector portion, and the arms recognize the foreign agent. That's how all antibodies are designed. We'll get to different flavors of antibodies shortly. Actually, almost an hour from now. Okay. How many antigens? Okay, what is an antigen? An antigen is the fancy word for the stuff to which an antibody binds. So if an antibody binds something, by definition, it's an antigen. It's a functional term. There's no, an antigen doesn't exist in nature per se, except when an antibody binds it, then by definition it becomes an antigen. So any chemical structure of any kind, any molecular signature that I've used that expression, that an antibody binds to is an antigen, period. Okay, how many antigens can that antibody bind? Someone else. That was a real question for the group. How many antigens can that antibody bind? Remember, here I'm an antibody, here's my trunk, and this is the part that communicates with the immune system. Here are my two arms, and there's an antigen binding arm and another antigen binding arm. So how many antigens? Come on. How many antigens can an antibody bind?
2.
2. Correct. Are the antigens? Yes. One antigen is bound here, and one antigen is bound here. So every antibody can bind 2 antigens.
Do you have to bind both for it to be like recognized, or can you bind just
We'll get to that shortly. Are the antigens that a given antibody, remember I said we have 1 trillion antibodies in circulation, so I'm going to pull one out of my body and hold it here. Are the antigens that this single antibody can bind, we know it can bind 2 now, the same or different? Are the antigens that a given antibody binds the same or different? How many people think it's the same? How many people think it's different? You're all right. Remember, I said the amino acid sequence of this heavy chain is identical to this heavy chain. The amino acid sequence of the light chain is identical to the light chain. Therefore, the structure that binds antigen from one arm and the other arm have to be identical because the amino acids are identical for all the proteins involved here. Therefore, the antigens that are being bound are identical. So this molecule is bivalent. It can bind 2 items, 2 identical items. It is bivalent. I think the next slide, I'm going to cheat and look and then go back. Not yet. Here's the same antibody in 3-dimensional structure. This is called this is taken from a crystallography picture. The color scheme is the same. Dark blue is the heavy chain. Light blue is the light chain. And it's the same Y-shaped molecule. And again, the FC, the effector portion of the antibody, is the trunk. Okay. A lot of times when I am talking to you, I'm going to talk about the antigen. There's an antigen binding part of the antibody, and it's going to bind antigen. And a lot of times I'm going to illustrate, and if we had a blackboard, I'd be drawing pictures all over the place, but we don't have a blackboard here, or if we do, I have to remove this. I'd have to go up and down on the screen. I have to talk to Heather about getting something here so I can draw. Anyway, a lot of times we're going to talk about the antibody binding the antigen, and I'm going to do an illustration like this. You sort of get the sense that there's a pocket at the end of the antibody, and the antigen fits into the pocket. And a lot of times, when I draw this course, I'll draw an antibody with a square pocket, a round pocket, a triangle, different shapes to show that different antigens fit in. That's a teaching tool. The message, however, is that it's actually surfaces. So even though I'm going to do this a lot and say the antibody bound an antigen, really what's happening is the antibody has a binding surface, and the antigen has a binding surface, and these 2 surfaces mesh with each other. So it's not a classic pocket. It's 2 surfaces meshing together. It's going to be, so here's a surface meshing with a second surface, and that's the antigen binding. See, you guys, I know you guys are really smart. You can actually look at this picture. You have all the information right now to look at that picture and tell me which side is the antigen and which side is the antibody. You actually can tell me from just the picture. Now the colors, you need to use the colors to help you. Anyone think they can do it? You all can do it. When I tell you how you know, you'll go, oh, of course. I just didn't see it. Which side is the antigen, which side is the antibody?
Would the antibody be the side with the two colors, and the antigen would have one?
Right. The antibody is the right-hand side because there is a blue and a red making up its surface. Antibody has a heavy chain and light chain. The antigen must be the yellow because there is only one color, yellow. So this is the light and heavy chain together make up that surface that makes the antibody specificity. And that's a very important message. I have never done this in class before, but I love it now that I just thought of it. The antigen pocket, even though I said there's no pocket per se, but the antigen binding domain, I haven't given it its formal name yet, is constructed due to the interaction between the heavy and light chain. So it's not like the light chain, you remember, again, the antibodies like this, and we got the light chain and the heavy chain, and they're running in parallel. It's not like the light chain binds the S of an antibody and the heavy chain of an antigen and the heavy chain binds the head of an antigen. It's not that. Together, the light and heavy chain together form a pocket. And so there's only one pocket per antibody chain, and that pocket is composed of a mixture of amino acids on the light chain and amino acids from the heavy chain. So even though the light chain and the heavy chain contributes to the pocket, there's only one pocket. Everyone got that? We good?
Is this only half of the antibody?
Excuse me?
Is this only one arm
This
of the
is,
antibody?
actually it's not even an arm, this is basically my hand. If I'm an antibody, this is just my hand that's grabbing the antigen.
Okay.
All the rest isn't even on this slide. Correct. So, even though I'm going to talk pockets a lot today, these are the kind of chemical interactions between those two surfaces that allow antibodies to bind antigens. So hydrogen bonds, ionic bonds, hydrophobic interactions, van der Waals, those are the chemical, those are just chemical, those four chemical interactions you learned in Chemistry 101, 5 years ago, whenever you took it. I never took chemistry, so I never learned it. And those interactions are what allow those two surfaces to bind with very high affinity. Antibodies bind their antigens with high affinity because of those chemical interactions. Now, hopefully this slide has what I want. No. I know the point of this slide, but it's silly. It's a waste of time. Okay, we're going to jump ahead and then jump back. There's an antibody, same Y-shaped molecule, but you'll notice now this is talking about flexibility. If an antibody were this, and only this, and I couldn't move my arms or anything, it would be a useful structure. But imagine if I can do this, 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 and that's what all these pictures are showing you. The antibody can flex. So not in this slide, although it's in this slide with BL and BC, but don't worry about it. A little bit more here. You notice that there's sort of a thing right here. Dark blue, light blue. Then there's another thing. Right? I'll give you a scientific word for thing in a minute. Right there. There's one there, there's one there, there's one here, there's one here. These are all flex points. So the antibody is actually constructed of, you see it here too. There's a domain. Thing is, the real word should be domain. There's a domain in the arm. So from here to here, there's a domain. Then from here to here, there's a second domain. Then there's a third domain here in the trunk, and a fourth domain. So each of those domains are their own structure, and they can twist and bend and wag and whatever all those words were on that slide. Rotate, whatever. Because a domain, dark green and light green, which is dark blue and light blue, or a different textbook, one domain, second domain, third domain, fourth domain. So antibody is made up of these domains, and the domains are somewhat structurally independent of each other, and there's going to be flopping around between them to give you antibody full flexibility. Good. One antigen combined here, the same antigen, same chemical structure, combined here. Why? And that is called bivalency. One antibody, bivalent for antigen. Why would you build an antibody with bivalency? I could have designed an antibody that looked like this. This would have been fine. One heavy chain and one light chain, all of that. I could have made my antibodies that, and we bound one antigen. Why is the antibody designed to be bivalent? What advantage? There's an advantage to us to have them bivalent. Any thoughts on why bivalency?
Efficiency in clearing the antigen
Clearing the antigen?
or recognizing it. I
What do you think?
don't know. I think antibodies being part of the immune response, if it's able to detect more efficiently.
You're close. I think you're definitely thinking in the right direction. So, let's talk about a little bit of chemistry. When we talk about adhesion, when we talked about adhesion, you know, the neutrophil going through the blood vessel and adhering to the endothelial cell and climbing through, there's an adhesion. There's an on rate and off rate. Any interaction that isn't covalent. So a covalent bond is, they're linked chemically and they cannot be separated. A non-covalent interaction has an on rate and off rate. The affinity, we're talking about high affinity. I mentioned that antibodies bind their antigen to high affinity. That's because there's an on rate and an off rate. Any kind of chemical interaction that is non-covalent has an on rate and an off rate, and the ratio of those two give you the affinity. So when an antibody binds an antigen, this arm of the antibody, have this one inert, binds an antigen, there's an on rate and an off rate. If the on rate is very high and the off rate is very low, you're going to get high affinity. But there is an off rate, so every once in a while, this antibody will let go of its antigen. By definition, the very interaction of an affinity means every once in a while the off rate kicks in and the antibody will let go. Bivalent. Did that little piece of information make you appreciate a little more why an antibody, what's the advantage of an antibody being bivalent? Didn't help.
So for the,
If it drops one, then it still has the other
Correct.
one as a backup.
What are the chances that the off rate is going to kick in at the exact same nanosecond for both sides? Pretty rare. So if the antibody has grabbed an antigen, or grabbed the chemical signature on a bacteria, remember our antigens are bacteria, they're not laser pointing. So the bacteria has thousands of antigens on its surface. An antibody grabs that bacteria, two antigens, the same antigen on that bacteria, every once in a while it'll let go of this one, but it's not going to let go of this one. And then it'll grab back on, and the antibody will never basically ever let go because of the bivalency. And I think the next slide, if I'm right, just says just that. Here's the bacteria or the fungus or whatever. Every once in a while there's an off rate, it lets go. But if it lets go, it's still hooked on the bacteria, high on rate, it'll grab back on, and the antibody never falls off, or rarely falls off, because of the balancing. Does that make sense, everyone? That's the design, why the antibody is what they are. Everyone good with that? Anyone confused? Good. Now, I emphasized, and I can do it in this slide, that there's a domain. Here's a domain. Heavy and light chain, light blue, dark blue, fold together to make a domain. Heavy and light fold together to make a domain. Heavy and light fold, heavy fold together to make a domain, another domain. In the very first picture I showed you, these domains were numbered, coming from the bottom going up, CH3, CH2, CH1. So each of those domains are sort of independent structures. And you can see it here in the three-dimensional structure. You can sort of see it here in this ribbon diagram of an antibody. And they're emphasizing here in a little more detail, is that each of those stretches, those domains I talked about, are 110 amino acids. You don't need to memorize the 110, but those 110 amino acids form a certain basic shape. And that shape is called the immunoglobulin fold. So that shape there on the left, notice the beta sheets, 1, 2, 3, 4, 5, 6, 7 beta sheets. That's an immunoglobulin fold. There are, if you look over here, a different part of an antibody. This is not the whole antibody. This is actually, this is my hand. This right here is my hand. Then this part here is my arm, and we don't have the trunk in this picture. But this immunoglobulin fold is of the heavy chain has an immunoglobulin fold that makes it an antibody, a component of an antibody. The light chain has a fold, and they fit together, and together they make those domains that I've been pushing. Why is that important? It will take us an hour to know why these immunoglobulin folds are important, unfortunately. So, a couple things to remember. All this will be on our exam type questions. Antibodies have identical heavy chains, 2 identical heavy chains, 2 identical light chains. The arms of the antibodies, called the antigen binding domain, they bind to different antigens, the trillions of antigens that are available. The trunk of the antibody, this portion called the FC, interacts with the immune system. It's bivalent, which means it can bind 2 identical chemical signatures, 2 identical antigens, and this immunoglobulin fold that I talked about. Each of those domains is made up of these immunoglobulin folds. Here's the flexing and bending and waving of an antibody. Here's why you have bivalency. And now, what is an antigen? Antigens can be carbohydrates, they can be lipids, they can be DNA, they can be small chemicals. They tend to be proteins most often. Not always, but the vast majority of antibodies target proteins. But remember, antibodies can bind any chemical signature. Right now, I guarantee you that I have an antibody in me that if someone went to Mars and brought back some chemical from Mars that we have never, ever, ever seen on Earth, I have an antibody that binds to that chemical. Now, how do we get that is the genetics we'll get to shortly, but in general, the vast majority of antibodies bind to proteins, but they can see any chemical structure of any kind. When they bind to proteins, they might bind to what's called the linear, I have to introduce the words. The part of the antigen, so here's an antigen, here's an antibody, and the Y is upside down here, so you're oriented. The Y is upside down, and the second antigen is not bound here, in this shape. So the FC portion here, here's the FC portion, and it's one heavy chain, the second heavy chain. Here's a heavy and a light next to each other. Here's a heavy and a light next to each other. So the Y is upside down. Here's a single antigen binding to one antigen binding domain. But the whole antigen doesn't bind to the antibody, only a couple of amino acids bind. The part of the antigen that binds to the antibody is called an epitope. The part of the antibody that binds to the epitope, meaning what on the antibody, remember those two, I showed you those pictures when I asked you about the colors, you had 2 surfaces interacting, it's not a pocket, it's 2 surfaces interacting. The surface that interacts with the antibody when it comes from the antigen is called an epitope, as written up there. The surface on the antibody that interacts with the epitope is called a paratope, Paratope. And I may put that on the exam. It's not important to memorize those 2 words, but those 2 words allow me to teach more efficiently. So the part of an antigen that is bound by an antibody is called the epitope. The part of the antibody that does the binding is the paratope. So epitope right here, paratope right here. Notice that antibodies can either bind a stretch of amino acids right next to each other, that's a linear epitope, or the antigens fold, and the antibody might bind an amino acid, then bind nothing in the protein chain, and then bind another amino acid. 10, 20, 100, 1,000 amino acids later. So there's a conformational, this is called discontinuous, I think I like the word conformational better. If the protein is folded, the amino acids do not need to be next to each other in the amino acid sequence of that protein. They might be next to each other, as shown here. They might not. And the structure of the protein matters. Because if this protein bends differently, then that amino acid is no longer near this amino acid, because the protein bends differently, that antibody couldn't bind it anymore. Okay, now we gotta figure out, what we haven't told you is, I've talked about the domains, I've talked about the immunoglobulin fold, I've talked about the shape of an antibody, I still haven't told you what in the antibody allows it to bind antigen. And it ends up, it's just a couple of amino acids. So from here to here is 110 amino acids. And from here to here, well, let me make the elbows a break point. From here to here is 110 amino acids, that's one domain. From here to here is another 110. From here to here is another 110. From there down, another 110. But of those 110 amino acids that make up the light chain, and of the 110 amino acids that make up the heavy chain that make up this part, only about 10 or 20 amino acids of that 220 actually makes up the paratope. And the reason is the antibody cells have structure. We can't just have the amino acids between my fingers and my elbow being random. 90% of them have to have that immunoglobulin fold to make this an antibody. This antibody has to have structure. It's got to be able to bind things and bind epitopes, antigens, and it's got to be able to interact with the immune system. So it has to have a fixed structure. So most of the amino acids in this 220 amino acids are giving it the structure that makes it you are an antibody. And then you got 10 or 20 amino acids out here that actually bind to the antigen. Is that clear with everyone? For everyone? Okay. And that, how do we know that? Well, how did we get to that point? Back when antibodies were first discovered, people would sequence them. And remember, the vast majority of, from here to here, the sequence of every single antibody is identical. From here to here, 110 amino acids, heavy chain. Identical. From here to here, 110 amino acids, identical. So identical sequence, heavy and light chain from here to here, identical. But here, we sequence this part, and it wasn't identical, which is what we need. Because every once in a while, if the amino acids that are my fingertips change, then I can bind this shape, I can bind that shape, I can bind that shape, I can bind that shape, and on and on and on. If my hand changes shapes into a trillion different shapes, I can bind a trillion different chemicals. I'm keeping all this identical. Does everyone see that? We just have to change these amino acids. Now, how do we find out that that was the way antibodies work? Well, we sequenced them, and we found out that, wow, the amino acid here is almost always the same, 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 always the same. Then we run into a stretch of 5 amino acids that are different, antibody to antibody to antibody. Same, different for 10 amino acids. Same, different for 8 amino acids. That's the 20 amino acids I'm talking about that make up my hand, to give my hand every shape possible. Those 20 amino acids change in the 110 of the light chain, and those same, so 10 of them change in the light chain, 10 of them change in the heavy chain, and then we got 20 amino acids changing the shape of my hand, but the other 100 amino acids in the light chain, and the other 100 amino acids of the heavy chain are identical to give me structure called an antibody. Is that clear? Anyone in trouble with what I said? So those trillion of antibodies that we started talking about are identical in sequence from here down to my toes. They just differ in the 20 amino acids in my hand. And the same 20 that give me this shape, or the same 20 give me this shape, the different 20 give me that shape, and so on. So those 20 amino acids vary from antibody to antibody to antibody. Those 20 or so amino acids are changing. And we have a name for this. It was actually in the first slide, and I ignored it on purpose. It's not a great slide. It was the best slide to show it. Okay, I guess this one. The 110 amino acids that go from here to here is called the variable region of the light chain and the variable region of the heavy chain. Then from here to here is the constant region of the light chain. Now the light chain stops. Remember, there's only two domains for the light chain. 110 for light chain here, 110 here. These 110 amino acids are identical, antibody to antibody to antibody to antibody, for the light chain. These 110 amino acids are identical, heavy chain to heavy chain to heavy chain, antibody to antibody to antibody. They are constant. Here to here, 110, constant. Here to here, 110, constant. This 110 varies to allow for the various shapes and therefore the various affinities. So this part from here to here is called the variable region of an antibody, and this part from here to here for the light chain, or from here all the way to my toes, is the constant region of an antibody. So those trillion antibodies that are in circulation right now, they are only varying in the amino acids of my hand, and the only variability is in the variable region. Variable region here. So I covered that. So, some refreshers, some points for the exam, and so on. Bivalency optimizes the fact that the antibody will never let go. Never letting go is called avidity. Antibodies are flexible. They can bend here, they can bend at my shoulder, they can bend at my neck, they can bend at my waist, in all directions. If it's a protein, the protein can be a stretch of amino acids next to each other, or they can be amino acids that are very, very far apart as the protein bends and curves and fits into the pocket. The region on the target, on the antigen, is called the epitope. I did write it down. Good. The region on the antibody, so the epitope on the antigen binds to the paratope on the antibody, and that's the antigen bind. So here's the epitope, and here's the paratope. And that is what gives an antibody specificity, because this paratope changes from antibody to antibody to antibody, and therefore the epitope will change as well. Okay, now we're back to 20,000 genes. How do we convert 20,000 genes into a trillion different proteins, all of which then have a different amino acid sequence? And we do that by something that is absolutely foreign to everything you've ever learned. How many folks have taken a molecular biology course or genetics course have some idea that DNA exists? So only half of you. That's not good. What more to do? Okay, you must have learned in school, you should have learned in school, that the DNA sequence in the cells that make up a muscle, the DNA sequence in the cells that make up a nerve, the DNA sequence in the cells that make up sperm are identical in every individual. You have a 300 times 3 times 10 to the 9th nucleotide in your body. It's a DNA sequence, and those 3 times 10 to the 9th nucleotides, that sequence is you. You've all heard about 23andMe. You get your genome sequenced. You pay $100 or $200 to 23andMe, you get part of your genome sequence. That genome sequence is identical in every single cell in the body. That is the hallmark of molecular biology. Except the cell that makes antibodies, which is called a B cell, threw that rule out. And the DNA sequence in a B cell is not the same as every other cell in the body. And that ability to change its DNA sequence allows a cell to make a different antibody from the cell right next door. So if B cell 1 changes its DNA sequence, it's going to make antibody 1. If B cell 2 changes its amino acid DNA sequence a little bit, it's going to make antibody 2. B cell 3 is going to change its DNA sequence yet again a different way. It's going to make antibody 3. We can get 1 trillion antibodies from 1 trillion B cells because each B cell changes its DNA different from germline, from the DNA that is us. The idea that a DNA sequence of a cell changes in a human just blew everyone. No one can accept it. That is not what happens. What is absolutely sacrosanct in genetics is you have one sequence and you have one sequence, you have curly hair because you have a sequence, and I have no hair because I have a DNA sequence, and that is it. But it ends up my B cells, every one of my B cells, change DNA sequence a little bit to make a different antibody. I have to emphasize here that the changes in the DNA sequence aren't everywhere. We've got 23 chromosomes. The only changes are in the heavy chain gene and the light chain gene. So we only have one heavy chain gene. All this bullshit I've been giving you about we must have 1 trillion genes, we only have one heavy chain gene. But B cell 1 changed the sequence of heavy chain gene a little bit, the DNA sequence. B cell 2 changed the sequence of the heavy chain gene a little bit, different from B cell 1. B cell 3 changed the DNA sequence only in the heavy chain, and its DNA sequence is different from 1 and 2. And, by the way, the light chain does the same thing. So the DNA sequence in a B cell for the light chain is unique. One B cell, one DNA sequence, one light chain. One B cell, one DNA sequence, one heavy chain. Still only one heavy chain gene, still only one light chain in quotes. I'll change that in a minute. But conceptually, there's only one light chain gene, and the B cell changed only the DNA sequence in the light chain. Changed only the DNA sequence in the heavy chain. And it's not huge changes. Remember, look at the antibody. This sequence is identical. This sequence is identical. This sequence is identical. So it's just changing the DNA sequence to code for those 20 amino acids that are changing to give us every shape possible. Let's stop a second. See if we're all okay. So now we need to find out how the hell can a B cell change DNA sequence, which goes opposite everything you've ever learned about molecular biology. Everything you've ever learned about genetics gets thrown out by the B cell, only in the heavy chain locus and only in the light chain locus. If I sequence the genome of a B cell in every other chromosome, it's identical to the sperm, the muscle, the neuron, the fibroblast, and so on. But the heavy chain sequence has changed. The light chain sequence has changed. So now the question is, how does it do it? You good? I'll stop for a split second. Any questions? Then now it's how. So we move into the how part. It ends up that there is the gene. Remember I said you only have one light chain gene. It's going to change its sequence, but the gene is still the gene. You're only going to have one heavy chain gene. And that gene is there, but it's going to change settings. It ends up that the light chain has a region within the gene called the V region and a J region. The V region is going to be for variable. So remember the fact that my fingers change amino acids in order to get every shape possible? Somehow that variable region has to change a little bit to give us DNA, new DNA for every amino acid combination. J contributes to this change a little bit, and we'll get to that in a moment. The heavy chain does it a little differently. The heavy chain has a region, another region, and a third region that changes the DNA sequence. So all the DNA sequence changes in the light region, light chain is going to be in blue and red. All the rest of the genome for the light chain is identical. From person to person to person, and from antibody to antibody to antibody. The heavy chain, everything in white is identical, no changes in the DNA sequence, which means no changes in the amino acid sequence. But there are changes in blue, whatever color this is in red. Purple? I can tell what color it is. Still don't know. Purple. So V stands for variable, because we want to vary the amino acid sequences. D stands for diversity. We have an antibody that has the diversity to bind everything in the world. And J stands for joining. Doesn't really make sense. Okay, within V, we have little chunks of DNA called V1, V2, V3. And within J, we have little chunks of DNA called J1, J2, and J3. So if a B cell takes V1 and moves DNA around and puts V1 next to J5, you're going to get a certain amino acid sequence right there, right? If the next V cell takes V2 and puts it next to J1, you're going to get a different amino acid sequence. Because V1, V2, and V3's amino acid sequences are a little different. The DNA sequence is a little different. The amino acid sequence that goes with it is different. The J's are a little different. So if we mix and match a V with a J, we're going to get one DNA sequence. If we take a different V and a different J, we're going to get another DNA sequence. If we take another V and another J, we get yet another DNA sequence. So by picking and choosing chunks of V and chunks of J, I'm going to end up with a different amino acid sequence right here in the pattern. Okay, yeah.
How many amino acids, like, roughly are these?
Excuse me?
How many amino acids, roughly, are these segments, like V1, V2?
The J is 20 nucleotides, so 3 into 20, 7 amino acids. The V regions are 10. The calculation gets a little complicated because part of the amino acids don't change in V to give and you still need structure. I still have to be an antibody. So all these amino acids can't change. So the V region actually is on about 110 amino acids, 330 nucleotides. But the change, V1 is different from V2, only in about 20 nucleotides. Okay, so if I take a random V and put it next to a random J, I'm going to get a bunch of different light chains. Everyone go with that? I don't have a good picture here, but if I take a random V, a random D, and a random J, here's the answer you decide. Okay. If I take a random V, a random D, and a random J and put them together, there are about 5 or 6 Ds or 5 or 6 Js or a certain amount of Vs. We'll get to that in a minute. I'm going to get a different heavy chain every time I put V next to D next to J because the sequences are different. So let's see if the numbers work out. The arithmetic is the following. This is mouse or human. It's mouse and human both. Don't worry about kappa and lambda yet. So let's do heavy chain. It's easier. VH, D. So remember the heavy chain is V, D, J. So VH for V heavy, D, could say DH, but there is only one D, and JH. Ends up we have 38 or so V's. Let's make it 40, it's easier. We have 40 V's, we have 20 D's, and we have 6 J's. Well, if you multiply 40 by 20 by 6, whatever that number is, that's how many different antibodies you can make. By just grabbing V1, you put V1 next to D1 next to J1. That's antibody 1. V1 next to D1 next to J2, that's antibody 2. V1, D1, J3, that's antibody 3. Blah, blah, blah. 40 times 20, 38 times 23 times 6. That's how many antibodies you can get just because you pick and choose your V, your D, your J. Okay, do you think 40 times 20 times 6 equals a trillion? Nowhere near it. So we're moving in the right direction. We've gone from one gene to 40 times 20 is 800, and then 10 more, 8,000. Now we have 8,000 antibodies. By just doing this, we have 8,000 antibodies. So we're moving in the right direction. We're moving toward a trillion. For light chain, 40 and 5. So we have 80, 200. 200 different light chains we can make. We can make 8,000 different heavy chains. We can make 200 different light chains. Remember, light and heavy make the paratope, not each one alone. So 8,000 heavies, 400, what did I say, 40 times 5, 200 lights. Put them together, and now 8,000 times 200 is 160,000, 1.6 million, I don't know, some number. It's not a trillion, but we're moving in the right direction. Because a B cell chooses this mixture of heavy chains, then another B cell might choose the same mixture, but it's not going to choose the same mixture of light chains. So when you put the heavy and the light chain together, you end up with another antibody paratope. So now we're up to 160,000 different antibodies. You choose V, you choose D, you choose J randomly, you put them together in the heavy chain. You choose V and J in the light chain, you put them together. One heavy, one light, put together. 160,000. We just created 160,000 different antibodies from two genes, heavy chain gene and light chain gene. Did I go too fast? Did everyone get that? Because 160 is better than 8,000. 8,000 is better than 1. 160,000 is better than 8,000, and we're moving toward a trillion. But we haven't quite got there yet. We will. So does everyone see the numerology I just went through? Anyone troubled by it? Okay, remember, there's only one heavy chain gene, but 40 V regions, 20 D regions, 6 J regions. Okay, one lie that I gave you, it ends up we have two light chain genes. We have a gene called kappa and a gene called lambda. So that 160,000 now becomes 320,000, because the B cell might choose to use kappa as its light chain, or it might choose to use lambda as its light chain. So we're up to 300,000 now, if I multiply that. It's either 300,000 or a million, but 3 million, but still we're not there yet. So two light chain genes, where you choose a random V and a random J. One heavy chain gene, you choose a random V, a random D, a random J. You multiply all those numbers together and you're up in the hundreds of thousands of different antibodies. This is really what it looks like. Remember I mentioned there's a kappa light chain and a lambda light chain, but on one heavy gene, and a bunch of 14 Ds and 40 Vs and a bunch of Js, and you multiply them all together. So that's what the chromosome really looks like. And then remember, all the sequence from here to here, and from here down is the same. That's what these constant regions mean. All the light blue constant regions. Those amino acid systems, this futzing around, or moving DNA around, it doesn't happen from here to here, or from here to here, or from here to there. It only happens in that first 110 amino acids. Because that's the only place where we're going to have different shapes, different paratopes. Same thing. How they did it, I am not going to do that to you guys. Not going to worry about that yet. Here are some other numbers, different textbook, ballpark the same numbers. And here's the arithmetic. Actually done for us. 41 times 23 times 6 is this number. 41 times 5 is this number. That number times that number. But there are two of them. Remember, there's a lambda chain and a kappa chain. We're up to 2 million. So just picking and choosing V, D, and J, picking and choosing B and J, having the two light chain genes, having a combination of any random heavy with any random light, you're up to 2 million antibodies. We're still way short of a trillion, but we're pretty impressive numbers. It's only 3 genes. Light chain gene 1, light chain gene 2, heavy chain gene. But the radical point here, and I want you to appreciate it, is the B cell took DNA, moved it around, and through the DNA, something I didn't say, and I have to say it, it's not important for immunology, but I have to say it because I am a molecular biologist. When V1 combined with J5, all the DNA between them was thrown away. This is the radical part of what we're talking about. The DNA sequence DNA was thrown away. That B cell will never ever have that DNA again. And that is the radical part, that a gene can be tossed, or chunks of DNA can be thrown out. But in order for V1 to be next to J1 in the J5 in the chromosome, V2, V3, V40 had to be thrown away. The DNA for that. And J1, this is J5. J1 through J4, the DNA had to be thrown out. It took people a long time to accept that was real. Okay. Let's go back to the easy picture to see it's easy. Remember, DNA codes for protein. They're triplets. So 3 nucleotide sequence, you get an amino acid. 3 more, you get an amino acid. 3 more, you get an amino acid. If you bring V1 and J5 together and you don't make it just right, you're going to be out of frame. If you have one base too many, it's 3, 3, 3, 3, 3, 3. If I bring V and J together and I slip an extra nucleotide in there somehow, now it's 3 for an amino acid, now it's these 3, and these 3 make a junk amino acid. I mean, they make a real amino acid, but it's not an amino acid that's going to be useful. And then the next 3 are another amino acid that's wrong. So V and J better come together perfectly. Otherwise, we're going to have frame shifts. And once you have frame shifts, you have junk proteins. That B cell is making junk antibody. That B cell is useless. It doesn't keep us alive. So that VJ connection has to be really clean. Perfect. How do we do it? We do it this way. So we have these things called recombination, rearrangement, what's RSS stand for? Signal. Rearrangement signal sequences. It ends up at the very end of V is in blue still. There is this unique sequence that's shown right here. At the very beginning of J, there's this unique sequence shown here. Those two sequences find the other. Well, they don't find each other. Proteins bind to these sequences. Proteins bind to this sequence. Proteins bind to this sequence. The proteins align with each other. That allows the DNA to align with each other. The nucleotides come in and cut and paste and splice and ligate and which we don't worry about today. And at the end of the day, you end up with a perfect V3 coming to a perfect J3. Because the proteins that bound to the, what's that, orange? Puke green. The proteins that bind to puke green next to V3, other proteins bind to brown next to J3. Those proteins find each other. Puke green and brown align. Cuts are made. And a perfect V3, J3 splice religation is made. So we haven't lost the codon triplets. We got that? Because, and if you look at this diagram, don't do it today because you have an exam, but if you look at this diagram, you'll see that because of the colors and the blah, 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 the perfect, only VJ can be made, only VJ, it all happens great. Because with just 2 of those, puke green and brown sequences, you can end up with VDJ in the heavy chain, VJ in the light chain, and it goes perfectly with no mistakes. Just, you'll have to look at this later to convince yourself of it. But it all gets together just perfectly. And there's BDJ, B and D and J come together first, D4 and J6, and then B18, J4, D6, J4. They all come together because of this alignment with these recognition sequences. So everything has got to be right in frame in order for all this to work. Everyone really comfortable with what I just said about triplets, alignment, perfect annealing of the end of B and the beginning of D and the end of D and the beginning of J. Absolutely have to be perfect. Everyone got that and understand why? Because I like you. It ends up, we're only at 1 million antibodies and we need 1 trillion. We need another trick. It ends up, this goddamn system purposely makes mistakes. So when B comes in and J comes in, they do come in perfect. Then nucleotides come in, nucleases come and chew away a little bit of B. And nucleases chew a little bit about J. And then put them back together again. Other polymerases come and add to B. They make it a little longer. 4 or 5 nucleotides. Adds to J, makes it a little longer, and then puts it together. So this nice, smooth joint that I've been trying to teach you about, to keep the triplets in frame, it isn't done. They purposely make that joint sloppy. And it's that sloppiness that gets us to 1 trillion antibodies. Because now, you don't know how many nucleotides are going to be thrown away from the end of B. How many nucleotides are going to be thrown away from the beginning of J? How many nucleotides are going to be added back to B? Past the end of B, just dangling loose. How many nucleotides are going to be added in the front of J, dangling loose? No template. Everything you learn, oh, that's another rule. Everything you learn about DNA, you copy, you got a C, you copy a G. You've got a T, you copy an A. When these nucleotides are added at the end of B and the beginning of J, there's nothing to copy. They throw in a C. They throw in 2 C's and a T. Or maybe they throw in an A, a C, and a T. And same at the beginning of J. So you randomly destroy the end of J, V. You randomly destroy the beginning of J, and then you throw in nucleotides out of the blue. That's how you get to 1 trillion antibodies. Because of the randomness of chewing back in both directions, and the randomness of adding nucleotides that have no reason to be there. The enzyme decided I want to add 3 A's, so it adds 3 A's. The next B cell, the enzyme decides to add a G and 2 C's, so it adds a G and 2 C's. The next B cell adds only 1 G. On and on and on. That's how you get to 1 trillion antibodies. Does everyone see that?
You said the polymerase would add, and what was the nuclease doing? Was it taking away, or they're both adding?
The nuclease is taking away from the end of B and the beginning of J,
That's what's
and the
them, right?
polymerase is randomly throwing on without a template. It's not copying as we've learned about DNA from day one. Why is DNA double-stranded? So Jane Watson can be a bigot? I mean, sorry, for those of you who know the joke, great. If not, I'll talk to you offline of that joke. It's not a joke. But everything about DNA is double-stranded, so you can copy. So every cell in your body has the exact same sequence because you've got something to copy. I'm just telling you there are enzymes that throw in random DNA, random nucleotides, without copying a damn thing. That's how we get to 1 trillion. Anyone have a problem with everything I just said? There's a major problem, molecular biology problem with everything I just told you. You may not get it. I'll tell you, you'll get it when I tell you. I don't think it's a hard concept, but I'm just hoping one of you. Let me give you a for instance. clean V, clean J, they come together, we've got our triplets perfect, right? Let's say there's no degradation of V, no degradation of J. They come together, we're perfect. What if that that polymerase adds one base? I said it can add any number, and it can add any of the four, it can add any number. What if it adds just one? We've got perfect V, perfect J, and now we've got one extra nucleotide thrown in.
They wouldn't.
That is allowed, that does happen. But what has happened? What's the problem?
They don't come together perfectly anymore.
And what is the triplets, what do the triplets look like now? They're fucked up. The triplets were fine when perfect V and perfect J came together. Once I added that one nucleotide, here's a triplet, here's a new triplet. It makes no sense. The next triplet makes no sense, the next one makes no sense, and the next one makes no sense. So the good news is we can get to 1 trillion antibodies. The bad news is 2 out of 3 genes you make is garbage. Because if you didn't add everything in factors of 3, 0, 3, 6, 9, you've made a junk protein. You okay? You look confused.
I don't know why it's so random.
To get to 1 trillion.
Why?
We need 1 trillion antibodies to protect us from every chemical signature out there. And this is, we need that randomness to add we need new amino acids. In every single finger, we need a new amino acid. The only way to get them is just randomly throw in nucleotides, and that's going to give us a new amino acid, another new amino acid, another new amino acid.
So do we have antibodies floating around our system right now that code for stuff that we've never been exposed to?
Correct. That's why I said I have an antibody against some protein in Mars or some chemical in Mars that we'll never see. I don't think I'm going to Mars before I die. I have antibodies. I guarantee you recognize chemicals that are on Mars, and so do you. Think about from a disease state. You have antibodies against the bacteria before you got infected. If God forbid anyone gets Ebola, some of you have antibodies against Ebola before Ebola got into you. How did that happen? By this randomness I'm talking about. Okay. Are we okay with why we have randomness, and you're okay with 2 out of 3 random events is a waste of time? It is a junk protein, or a junk gene, which leads to a junk protein. All of this is happening on the heavy chain, both with B and D, and with D and J. So that means with heavy chain, I think the numbers are 8 out of 9 genes are going to be garbage. 2 out of 3, 2 out of 3. I think it's 8 out of 9 genes with heavy chain, because you're doing this randomness twice. VD and DJ. So 8 out of 9 attempts in the heavy chain to get a new hot, Rodeo Drive quality antibody. 8 out of 9 attempts is junk. With the light chain, it's only 1 out of 3. 2 out of 3 is junk. So the trade-off is randomness and therefore diversity with junk. And that's what the system chose. Yeah?
So is the premise of that just that we need to excise and replace nucleotides in triplets?
No, if we had enzymes that replaced them in triplets, we wouldn't generate junk. But we don't. We have this system instead.
So when it does happen to replace it in a triplet, that's that 1 out of
Right.
5.
If it took 3 away from V,
Yeah.
and took 3 away from J, and they added 6 or 3 or 9, everything's kosher.
Okay.
But there's no rule as how many you should add, because there's no DNA to copy. The enzyme is throwing in this nucleotide. So that's how we get, so this is a list of the enzymes that are involved in doing all that craziness that I just talked about, the randomness that I talked about. Don't have to worry about it. But we do have to talk about this slide. I'll make it simpler. And I want you to appreciate this. The heavy chain is rearranged first. you go to the heavy chain gene, 8 out of 9 times you're going to get a junk protein. 1 time you're going to get a good protein. What are you going to do with those 8 B cells that make it a junk protein? What would you do with them? Any ideas? What would you do with the 8 out of those 9 B cells? We're going to make a junk heavy chain. What would you do with that B cell? We'd kill it. There's nothing else to do. It's moved the DNA around. It made its choice. It created a junk protein. We're finished. It's finished. But how many of you have both a mom and a dad to create you? Were any of you created without an X and a Y or 2 X's coming together to create you? I don't believe so. So how many copies of the heavy chain gene locus do we have in a B cell? In every cell, we're talking about B cells here. We got a heavy chain locus from mom, and we got a heavy chain locus, we're diploid. We got a heavy chain locus from mom, we got a heavy chain locus from dad. Everyone okay with that? It ends up the B cell has 1 more bite of the apple. If it goes on to mom's chromosome and screws up 8 out of 9 times, it shuts that chromosome down, goes to dad's chromosome, dad's locus of the heavy chain, and tries again. So you get 2 bites of the apple to get a good heavy chain. Did I lose anyone? Really, really important. I'm going to add 1 more rule. Did I lose anyone? Why it's 2 bites of the apple? What if you go to mom's chromosome first, and it does work? You've got a good heavy chain. What do you want to do with dad's copy? Anyone want to take a guess? You don't want to touch dad's copy. You've got yourself a good heavy chain. The chances of getting a good heavy chain are very slim. 1 out of 9. 8 out of 9 times you're going to screw up. So if mom, you rearrange mom with all this randomness, and it worked. You don't want to touch dad's. You want to shut dad's off and never ever look at it again. Does that make sense, everyone? So you have 3 options. You rearrange mom, it works. You shut off dad. You rearrange mom, it fails. You go to dad and you just rearrange dad and you hope it works. If dad rearranges and it's okay, bingo, you have a heavy chain. If dad fails, you got a dead B cell. Did I lose anyone?
Does it always start with the...
Oh, no, mom and dad was for teaching purposes only.
Okay, so
It chooses a chromosome, tries, and then either does or doesn't choose the other chromosome. Okay, I gotta give you a word. It's on the slide. It ends up that you have 2 copies of every gene because of mom and dad, except for X and Y of your other chromosome. Those are called alleles. Mom gave you a heavy chain, it's mom's allele. Dad gave you a heavy chain, that's dad's allele. The textbook is a little more sophisticated than me, and actually calls them allele 1 and allele 2. And this chart is just telling you what it means there is you go after heavy chain, you get a productive allele 1. If that's the case, you don't touch the other chromosome. The second allele is excluded. And here's the dead one. You try with allele 1, you fail. Non-productive, you screw up. 8 out of 9 times you'll screw up. You try with allele 2, 8 out of 9 times you're going to fail. Now you kill the B cell. And this chart is just taking you through all the various places to end up with an antibody. So after you finish heavy chain, if you can't get a good heavy chain, you're dead. If you get a good heavy chain, now you got to start on the light chain. The good news about the light chain is you get 4 bites of the apple. Remember, there are two light chains, one from mom, one from heavy. One from mom, one from dad. You've got that second gene of light. One from mom, one from dad. So you get 4 tries with the light chain. And one of these guys is going to die. You try 4 times, you still could die. Because 2 out of 3 tries with the light chain is going to fail too. So 2 bites of the apple for heavy chain, moms and dads, professionally allele 1, allele 2. 4 bites of the apple, 2 genes, 2 parents, 4 bites. All of this is occurring in the bone marrow. So the bone marrow is a killing field for B cells. Because the numbers are working against us here. But all of this is going on in what's actually called a pre-B cell, but don't worry about that. Heavy rearranges. Mom's heavy rearranges first. Dad's heavy next. Or dad shuts down. Kappa moms. Kappa dads. Lambda dads. Lambda moms. 2 bites for the heavy chain, 4 bites for the light chain. You either die or you don't die. The B cell either dies or don't die. At the end of the day, you have an antibody that looks like this, with a very unique protein amino acid sequence right there in the paratope, or you have a dead B cell. Key points. This rearrangement, moving V and J around, V, D, and J around, choosing V at random, choosing J at random, choosing D at random, all that's at random. V, D, J for heavy, V, J for light. Then some small number of nucleotides are removed and added at the junctions. More randomness, more possibility for different amino acids in the paratope. And then allelic exclusion means once you've got something good, you don't work with the other allele, or if you generate garbage from the first try, you do go to the other allele. That's what all this allelic exclusion means. And your goal here is to create a protein that remains in frame. With this random removal, random choice of V, random choice of J, random choice of V, random choice of D, random choice of J, random removal, random addition at all those joints, and the numbers work out. Because of time, and I really wanted to give you guys a breather, about 10 minutes. Yeah, I can do 3 minutes and then 7 and we'll be out of there. It ends up we make 2 antibodies. When the B cell finally figures out what to do with the variable region, it actually ends up making 2 antibodies. It makes 1 antibody that has a little tag on it and it gets secreted. The antibody, the B cell releases the antibody, the antibody goes into blood, and the antibody is there to protect you if you need. If the right bug comes along, the antibody will bind to that bug and you're protected. The antibody also retains a copy of the same antibody. The B cell retains a copy of the antibody as well by inserting it in its membrane. You'll understand why in about 2 minutes. But the B cell doesn't want to lose contact. We need, if you've made an antibody that is anti-Ebola and you get an Ebola infection, we want more of those B cells. You know, you've got 1 B cell making that 1 antibody against Ebola, great. That's not enough antibody if you actually get infected. You want 1 million B cells making that 1 same antibody. So you've got to get the message back to the B cell. This antibody you made, 3 years ago, it's fantastic. We need 1 million B cells to make 1 million, billions of that antibody to keep us alive. So the B cell can't lose contact with the antibody that it made. So it retains some antibody on its cell surface so that if you ever need to call on that B cell because it made a really good antibody, you can find it through that antibody held on the cell surface, the membrane now forming antibody. Is that okay with everyone? I won't tell you how, I was going to tell you how, because we're going to cut the class short, I'm not going to tell you how the B cell makes these 2 antibodies. But the reason it makes the 2 is important. This is a secretion looking for bad guys. This is retained so that we can find the B cell that made a useful antibody if we ever need to find it. Anyone lost? No? I'm not going to tell you how. I'm more than willing to tell you offline, but not now. Call me, come by my office, and I'll tell you how. It's on the slides. I'm cutting some stuff out for you guys. And that antibody retained on the surface is called the B cell receptor. I think that's the only. Yeah, the first bullet means the 2 antibodies, one secreted, one retained, and the one that's retained is called the B cell receptor because we want the system to be able to find that B cell. We have a need to use that B cell to be found. Last thing. Remember, variable region, constant region, constant region, constant region. Those light blues, I lost it, right here, are constant region. They're close to the constants. So here's the VDJ to get variable, to get all the paratopes we need. And the polymerase, the RNA polymerase is running along, copies VDJ, then copies these mu1, mu2, mu3. So the closest constant region is mu1 through mu4. Mu, you'll need to learn some Greek alphabet. Mu is M, alpha is A, delta is D, epsilon is E, gamma is G. You just have to memorize the 5 Greek letters because when we talk about gene, we talk Greek. When we talk about antibody product, we talk American or whatever language we speak. So it ends up we have 5 different antibodies. Call those 5 Greek letters. And here's the Greek letter for the gene. Here's the antibody name. And it ends up the first antibody a B cell ever makes is an IgM. Because M comes from the mu gene. And if I showed you that picture, just to remind you, the mu gene is the first gene in the chromosome. So every B cell that pops out of the bone marrow makes an IgM because of this genetic design right here. But that B cell has the capacity of making an IgG, an A, an E, or a D. That's the other reason that's the other reason the B cell retains some antibody in its surface. If we have to go back to that B cell and say, you know what, IgM was good, but if you make an IgA, your host is going to be a lot healthier. So change from an M to an A. Well, we need to find the B cell that made the antibody that kept us alive for a little bit to make that switch from M to A. So we have to find that B cell. We're connected to that B cell by this B cell receptor. Does everyone see that? How it all sort of hangs together, hopefully hangs together for you? So, the B cell, after the antigen, after the infectious agent enters, about 7 minutes, after the infectious agent enters, cells will, the adaptive immune system through T cells, which we'll hear about tomorrow, the adaptive immune system will decide, you know what, an IgA is a lot better than an IgM, for reasons I'll tell you in a minute. So you go back to that B cell and you say, stop making M, start making A. But we have to find that B cell. Because every B cell, remember B cell 1 is making anti-Ebola, B cell 2 is making anti-flu, B cell 3 is making anti-strep, B cell 4 is making anti-staph, B cell 5 is making anti-Martian. You got to find the right B cell. So stop making M and start making A. We only want the B cell that's making anti-strep to make IgA. We don't want the B cell that's making anti-Martian to make IgA because we have no Martian antibody, no Martian antigen right now. That? Oh, so here are the 4 or 5 antibodies. Ends up, IgM is actually a pentamer. Oh, a little math for you before you go home, before you start studying for your exam. How many antigens can an IgM bind? IgM's a pentamer. There are 5 IgMs here.
10.
Excuse me?
10.
10. 1, 2, times 5. So IgM can bind 10. IgA is a dimer, it can bind 4. These guys can bind 2. But what's really important, and as IgM is a pentamer, IgE you'll hear about when you talk about allergy, hopefully. If not, we'll talk about it. The B cell, I won't tell you how, but the B cell, one more time, when it's making IgM and now it means IgA, it throws away all the DNA between the M, the mu, and the alpha locus. Again, the B cell is throwing DNA away. This is unheard of until we heard of it. So, in order to make IgE here, here's mu, delta, gamma, 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 epsilon, alpha. We want this B cell to make IgE. All this DNA between mu and epsilon, we want this product, IgE. All this DNA in between, red through green, thrown away. Now why do we have, 5 antibody isotypes? There are their names. They're called antibody isotypes, they're called classes. So class switch is when the B cell decides to stop making M and start making something else. For class, we're ignoring D, don't worry about it. And the B cell has to retain a copy of the antibody in order to be found as it's useful. This, I gave you 2 slides on what the various antibodies do. This is the simpler slide. I really want to stop now. Let's talk, if you're willing to have me come back, it's your call. Let's talk whether you need to know anything on this slide after you learn hypersensitivity. I'm teaching you how the immune system works. You're going to get autoimmune, you're going to get an autoimmune lesson, you're going to get some oral immunity lessons from Dr. Weinberg, you're going to get some hypersensitivity, you're going to get pathology lessons coming up. And I don't know what the pathology people will teach you, and if they don't teach you what I think they should, then you don't need to know this crap. So we'll wait up here. And of course, if I don't come back, I will test you on this. But the main point here, let's just pick one thing, 2 things that you probably know about, so you can relate to it. We talked about complement, fixing complement. C1q will bind to antibody. It doesn't bind to all the antibodies, it only binds to which complement? Oh, complement. Notice that M is good for complement and G3 is good for complement and some of the other ones are not. So the complement system only uses some of these isotypes, not all. The other one is IgE. Mast cells. Allergies. If you have any of you have allergies, about 15% of you in this room, about 2 or 3 of you have allergies. Dog hair, pollen, ragweed, whatever. Those are all mediated by an antibody called IgE. So allergic responses are IgE. You may or may not get it from the faculty member, and we'll talk. If you do, we'll review it. So IgE is involved in an allergic response. So each of these play a different role in the immune system because the trunk of the antibody is what communicates with the immune system. When my hands find the antigen, the rest of me communicates with the immune system. Good luck with your exam, and thank you for coming.
Thank you.